Hello and welcome everybody. Today's speaker is Dr. Jahnavi Punekar. She received her master's degree from Indian Institute of Technology, Bombay, and her PhD from Princeton University. Her expertise include foraminifera, biostratigraphy, paleo-environmental, paleoclimatic, and paleoecological reconstructions, and geochemistry of major climatic and faunal turnover events in Earth's history. Today, she's going to talk about the End-Cretaceous Planktic Foraminifera crisis that may be linked to ocean acidification. So please take it away.
All right. So I'm going to put this on full screen mode. Thanks a lot, Prajakta, for such a nice introduction and for inviting me. It's an honor to be presenting at the LPI. The title of my talk is The End-Cretaceous Planktic Foraminifera Crisis and its link to ocean acidification. So before I begin, I would like to thank the funding agencies who have helped conduct this research. First off, Princeton University's Scott and Tuttle Funds and NSF Foundation grants, which basically funded my research during PhD, and the Department of Science and Technology India Inspire Faculty Award, which funded my research in the last couple of years. Before I begin, I would also want to express my gratitude to my mentors, Professor P.K. Saraswati from IIT Bombay and Professor Goethe Keller from Princeton University. My research focus and direction has been largely influenced by these two people. So let me start with, if you go to Google Scholar and just type in mass extinctions and play with the time filter, in the 1980s, you find but 3 papers about mass extinctions. If you change this time window to the 1990s, you find about 4 or 5 publications. Change this window to the 2000s, it's 3 pages of worth of papers on mass extinctions, more about the 6th mass extinction. And if you change this to from 2010 to 2020, I could count up to 30 pages, each with about 10 to 15 listings. So that's over 300 papers. So this kind of is a nice crude estimate of how the interest in mass extinctions and mass extinctions research has grown over the past 3 decades, and rightly so. Here's one of my favorite figures from Barnowski et al. That talks about a 6th mass extinction going on right now, anthropogenic and a serious biodiversity crisis. This figure is x-axis shows extinction magnitude in percentage of species, and y-axis in extinctions per million species year. It's one of the metrics for extinctions. And if you see the plots here, the bars, these, so you have Permian, Ordovician, you have Triassic, this is Cretaceous, and this is Devonian. So you see these bars, and that the extinctions played out on different time scales. If you hypothetically shrink these extinctions to, let's say, a 500-year time window, they plot as these dots here. This is just a reference, okay? And all of them are mass extinctions, which means the species loss is above 75%. All right, so if you look at the white, the yellow, and the orange dots here, these are actual data based on species already extinct in the last 500 years, critically endangered in the last 500 years, and threatened as of right now. Now, if you hold the extinction intensity constant and project this to 75% biodiversity loss, the estimates suggest a best case scenario of 200 to 500 years for the next extinction. And this was the worst case scenario. The best case scenario is about 900 to 2000 years. This is when we can expect the next mass extinction. just so that this becomes more relatable, this would range from, let's say, 3 generations of human beings to about 30 generations of human beings. So I think we are too close to a crisis to afford to be skeptical at this point. And it's really good to know that more people are working on understanding mass extinctions and the future of us as a species on this planet. I highly recommend these books, The Sixth Extinction, The Earth After Us, The Ends of the World. There are many more. These are popular science books, and I'm sure many of you must have already read these. Really nice to for people who are not technical, not too technical, but want to understand what's going on. All right, so now that hopefully I've impressed upon you the importance of studying mass extinctions, how do we define them? A mass extinction is basically loss of more than 70% species within a very short window of geologic time, which is basically accelerated extinction rates, extinction per time. These are associated with ecosystem collapse and some major geochemical cycle perturbation, could be carbon cycle, for example. And these are often linked to a catastrophic trigger, may not always, but most often are. And these catastrophic triggers could be extraterrestrial impacts or large igneous province volcanism. Mechanisms of extinction include abrupt climate shifts, extreme warming, carbon cycle perturbation, ocean acidification, and many more, sea level changes, and so on. My particular research focuses on this aspect of the Cretaceous-Palaeogene boundary mass extinction. So a brief introduction to the big 5 mass extinctions of the Phanerozoic, which is the past about 540 million years. The first one is approximately 440 million years ago, which records an extinction of 85% of the species that were present then. This has been linked with orogeny and related carbon dioxide drawdown, leading to extreme climate cooling, glaciation, and sea level fall. This is the End-Ordovician mass extinction. The next one is End-Devonian, dated around 370 million years ago, that witnessed 70% species loss. This has been linked with the colonization of land by vascular plants, leading to enhanced erosion and marine eutrophication and anoxia. The third one is at the end of Permian, around 252 million years ago, and this witnessed a major biodiversity crisis with 95% of all life forms going extinct. It has been dubbed as the mother of all mass extinctions and has been linked with mostly large igneous province volcanism, 2 of them being Emeshan traps and Siberian trap volcanism. The mechanism is greenhouse warming, marine acidification, euxenia, and so on. The next one is end of Triassic-Jurassic boundary mass extinction at around 201 million years ago, witnessed a species diversity loss of 76%. This has been linked with the Central Atlantic magmatic province volcanism and associated greenhouse warming and possible marine dysoxia and acidification. The last one and the most recent one of the big 5 mass extinctions is the Cretaceous-Palaeogene boundary mass extinction. And this is dated at 66 million years ago and witnessed a species diversity loss of 80%. Now, the hook for studying Cretaceous-Palaeogene boundary mass extinction is the demise of non-avian dinosaurs. This is interesting to kids as well as adults, and this is what makes this mass extinction so interesting. I've put in a collage of some of these artists' impressions of these non-avian monsters. The first one is a T-Rex. The second one, this is a T-Rex. This is a plesiosaur, pterosaur, ichthyosaur, mosasaur. But what we must note is that there is also a mass extinction seen in microscopic fossils, especially Planktic Foraminifera and Coccolithophores. Another interesting point that I would want to mention here is the marine cephalopods, the microscopic planktic forams, and the coccolithophores are all calcium carbonate secreting organisms. And this shows a pattern of calcium carbonate secreting organisms getting adversely affected by this event. I would like you to keep that in mind. So what makes the KT boundary, the KPG boundary, so interesting? If you look at this figure here, it's a nice summary. If you follow the arrows, this is the big 5 mass extinctions plotted here, 4 and 5, all the way from Paleozoic, Mesozoic, Cenozoic, older at the bottom, younger at the top. The first panel is extinction intensity. The second panel shows impact events in terms of their ages and also the size of the impact. The third panel shows you large igneous province and continental flood basalt province volcanic events. So what makes the KPG boundary unique is that you have a apparent coincidence of 2 triggers. One is the Chicxulub impact, which left a 180 kilometer wide crater in the Yucatan Peninsula, Mexico, and Deccan volcanism, which is in India, much of peninsular India. And aerially, this has been compared to the size of France. The amount of basalts that were erupted is, some of the estimates are 1.5 million kilometer cube of basalts in a very short geologically short time interval. So because of these 2 major catastrophic events in such a small geologically short time window, it makes the Cretaceous, Tertiary, Cretaceous-Palaeogene boundary mass extinction very controversial. And of course, you have people who like the impact more as an explanation for this crisis, and people who like the volcanism more. This is another figure. Let me just discard the changes and go back. Yeah, so this is a figure by Cortio and Wren from 2003. And as you can see, the x-axis shows ages of continental flood basalts. And this shows the ages of, the y-axis shows the ages of mass extinctions. And you see a pretty good correlation here, Deccan and End-Cretaceous being right here. Okay, so the controversy itself has lasted for over 3 decades, and I don't think I can cover that in 3 minutes or even 30. So I will steer clear of it and move on to my research. And I work with Planktic Forams. This is the tool of choice to reconstruct past environments and understand how this extinction played out. So you can see these images here. These are scanning electron microscope images of extremely small marine planktonic single-celled heterotrophic organisms which float around. You can see an animation playing here. So they are all the way in the surface of the ocean from surface to subthermocline depths. And when they die, the shells settle down to the bottom of the ocean and become part of the sediment record. Now, what's interesting about these organisms is they're very sensitive to the environmental changes, and they also evolve pretty rapidly. And the shape and the morphology of these shells and the species have an age context to them as well, which means that if you know what species indicates what age, you can actually determine relative ages of packets of sediment. The other good thing about these is that they are usually cosmopolitan, which means they are all over the world's oceans. And when they settle out, you can actually correlate distant sediment sequences based on what time indicating foraminifera are present in them. So this is very important for the kind of work I do. So with this introduction, let's talk about how do we get these forams? How do we decide where to find them? So we either go do field work in sediment sections, outcrops, we do field work, and collect samples at regular intervals, pre-decided intervals, put them in Ziploc bags, label them, bring them back to the lab. Or we order these cores from core repositories and then sample them and then process them. How do we process them? Break them down into smaller fragments, soak them in hydrogen peroxide to remove organic carbon, and then wash them thoroughly with a stack of sieves. The residue that we get, we dry in an oven and then archive in vials, and then spread it out on these micropaleontological trays and use a stereo zoom microscope to study them. So here's a small animation that I made for my students. So you have forams settling out from the surface of the ocean, and once they are part of the sediment record, we would basically go and collect these from different levels, the lower ones being older and the upper ones being younger in terms of relative age. Once we collect these, basically wash, dry, put them on the slide and study, use a brush to pick them, mount them in assemblage slides like this, and then we identify different species amongst them, and then graph this data up such that we get plots like this, which shows the relative abundance of different species, along with a lithological log representing where the samples came from. Okay, so this is just to orient you with the eventual data that I will show you and how to interpret those graphs. Quick recap, y-axis is relative time. Lower sediments are older, the upper sediments are younger. Okay, so let me introduce the Cretaceous-Palaeogene boundary from a micropaleontologist's perspective. There are three primary criteria to define the KPG boundary. First is the sharp lithological change with a thin red layer, which defines the KT boundary in field. The marls, and then the thin red layer, and then you have darker sediments, clay, and with recovery of marine calcifiers, there is increasing amount of carbonate in the sediments. This is a very general picture. More importantly, how do we define the boundary? We define the boundary based on the extinction. The extinction of Cretaceous Planktic Foraminifera, which are very ornate and intricate, and usually larger than the ones that evolve after the crisis, after the extinction. These are much smaller and simpler in their morphology. So the primary criteria to define the extinction is the extinction itself, the extinction of Cretaceous forams and the new evolution of Paleogene forams. There are secondary markers, however, such as 2 per mil negative shift in the delta 13C, and this could be bulk rock or based on species. And also the famous iridium anomaly, which is usually concentrated in the red rusty layer at the KT boundary. Now, that has become synonymous with the impact because Louis and Walter Alvarez, this is a nice picture of them from Gubbio in Italy, discovered this thin layer which had iridium. However, I would like to emphasize that the iridium anomaly does not define the KT boundary, but the extinction does. And interpretations will accordingly vary. So how do we use forams? We use forams. These are Planktic foram pictures here, and these are the age-specific ones that I was talking about. If you can identify these in sediments, you can basically divide these sediments into packets of relative time. And that's how we provide a relative age framework to events that are happening in the sediment succession. So what else do Planktic forams tell us? Here is a very nice picture by Abramovich and Keller. So borrowing concepts from ecology, Planktic foram communities which have high diversity, this is especially for the Cretaceous, optimum assemblages show high diversity and high abundance of these ornate Cretaceous species. With increasing stress, especially closing in on the KT boundary, especially in the stress intervals, you see that there is a diversity drop and an increase in the abundance of generalist species, and dominance of this particular species. The genus is called Guembellitria. It's a triserial planktic foram, and it has been also called a disaster opportunist because in samples where other forams don't seem to do very well, this guy is abundant and dominates the assemblage. All right. Similarly, for an analog for the early Paleocene, post the extinction, now the extinction level is here and time axis is here. You can see that with time, the survivors of the K-Pg boundary dwindle, except for the triserial foram that stays on for longer. It's the only long-term survival of the K-Pg boundary mass extinction. Also, with time, you will see an increase in the diversity and abundance of new evolved forams. All right. So based on this, we reviewed a lot of K-Pg boundary sections all the way from India to Israel, Egypt, and Texas, and found that immediately preceding the K-Pg boundary, we have abundance of these disaster opportunist forams indicating high stress. And also in what we call zone P1b, which is a time indicator, relative time indicator, we find again a dominance of this particular triserial planktic foram. And back then in 2014, we had tentatively linked these with episodes of Deccan volcanism, although we have much better estimates of the timing and duration of volcanic pulses now, which we will discuss eventually. So that brings us to recent developments in Deccan research. These, if you look at this inset figure here, these are red bowls. And a lot of attempts were made to extract zircon crystals for uranium-lead dating from the basalt itself, and those failed. But Blair Schoen's group from Princeton collected red bowl samples which occur intermittently in the basalt. And these yielded very nice zircon crystals, which could be dated using uranium-lead technique. And based on that, these levels show all the red bowl samples they collected across the stratigraphy of Deccan, and provided a very nice age framework, which allows us to understand the timing and the eruption rates. So now if you look at the figure on the right side, the x-axis shows the absolute ages, and the y-axis shows your cumulative volume of the basalt that's erupted. I would like you to focus on this plateau that you see here. What it means is within a short bracket of time, a lot of volume of basalt was erupted. And this correlates with one of the high volume eruptions. It's Poladpur Formation. And in terms of timing, this immediately precedes the age of the K-Pg boundary. So the biggest voluminous, possibly catastrophic eruption occurred immediately before the K-Pg boundary. So how does volcanism connect to biological crisis, and in this case, the K-Pg boundary crisis? One angle to it is the excessive amounts of sulfur dioxide injected into the atmosphere. These react with hydroxyl free radicals if they reach the stratosphere and form aerosols. These aerosols affect the absorption and scattering of sunlight, causing short-term cooling, and also acid rain on a decadal time scale. Acid rain on continents results in a dissolution event, and also enhanced silicate weathering, which can eventually result in mesotrophy in shallow marine environments. However, more importantly, it can cause short-term surface ocean acidification and a carbonate crisis for marine organisms. The other angle to volcanism is more serious, and this is excessive amounts of carbon dioxide injected into the atmosphere. And with higher PCO2, this would act on a millennial timescale and result in global warming. Air-sea gas exchange would result in a lowering of surface ocean pH, which would eventually result also in ocean acidification. Together, these can lead to severe biological crisis and possibly even extinctions. Now, this is a small inset figure from Honish et al. This was back in 2012. Um, this has very nice boron isotope data tracking pH changes through the Phanerozoic, and especially for the mass extinctions. Although they don't make a big deal about any acidification at the K-Pg boundary, you do notice that for planktic forams and for calcareous nanofossils, there is a major drop in the diversity and abundance. So those were affected. Benthics, not so much. Shallow reef builders, a little bit. So planktic forams and calcareous nanofossils did get hit at the K-Pg boundary. All right, so in 2011 and in 2014, Eric Font from the University of Lisbon presented some very unique and interesting evidence, which he called the chemical benchmark for Deccan volcanism. Now, this was an interval in Cron 29R immediately preceding the K-Pg boundary, and within what we call the Planktic Foraminifera biozone CF1, which amounts to, let's say, about 229 kilo years before the KT boundary. So this entire duration would be about 230,000 years. This is the latest estimate. So this forms an interval immediately preceding the K-Pg boundary, and he defined it on the basis of anomalously low magnetic susceptibility and the presence of an exotic mineral called akagenite, which is named after a mine in Japan called Akagen. And this is basically an iron hydroxychloride feathery platy mineral, which is supposed to have formed in the atmosphere with reaction of volcanogenic chloride with Fe2+. So that's one. And the second one was continental acid rain leading to continental dissolution of magnetite, resulting in low magnetic susceptibility once the sediment is deposited in the basin. So this led us to think about a possible acidification event in the surface oceans, which should have affected carbonate secreting organisms if this hypothesis was true. So CO2 from the atmosphere reacts with water to form HCO3 minus and H plus. This is the excess proton that causes the lowering of pH. Now, planktic organisms build their shells using CO3 2 minus and Ca2 plus. And what's interesting is that if you have more CO2 pumped into the atmosphere, this reaction would proceed in the forward direction, releasing more protons, which would react with carbonate ions to go back and form the bicarbonate ions. In this process, they deprive the calcifying organisms of the carbonate ion, and this is what results in the calcification crisis, in this case for planktic forams. Now, let me just clear this up for a moment. So, all said and done, if there is no low pH and no calcification crisis, planktic forams that live in the surface of the ocean would die and settle out at the bottom of the ocean, and with minimal damage, depending on the local settings. But if in the same settings you have lower surface ocean pH, the organism won't be able to build the same kind of shells and may build thinner walled shells. That's one possibility. The other possibility is that it might actually dissolve and become more vulnerable during its descent through the water column. In either case, it is rendered more vulnerable to bottom water transport and breakage, which means the same foram in a low pH setting would not have broken down as much, but will be more fragmented if there was a low surface ocean pH. In the same scenario, the benthic forams that live at the bottom of the ocean should not really get affected. All right. So another thing that can happen as an adjustment to such a calcification crisis is the organisms won't be able to build their usual sized shells, but will end up building smaller shells. And this is what we call intraspecific or within the species dwarfing. So these are the possibilities of what may happen to planktic forams if there is a surface ocean acidification event. In our case, if it's related to Deccan volcanism. So we went back to the same section that Eric Font had marked the Deccan benchmark interval at. And we basically studied the fauna, and the extinction looks sudden and there's not much happening, which was initially a little disappointing. But we focused on the interval, which is the last 60 centimeters of this section. And I'm reminding you that this is biozone CF1, and these are older sediments and these are younger sediments, and the red line is the KT boundary, and we're talking about the final 60 centimeters here. So what we saw was poor preservation of calcium carbonate. Now, poor preservation of calcium carbonate of shells is not unusual in planktic record, but in this case, it was just before the KPG boundary, and that made us want to study it a little more. So we devised a proxy because we need a systematic documentation of this event. We divided the foram preservation into 3 groups: the good, the bad, and the ugly. The good is perfectly preserved shells. The bad is imperfectly preserved shells with dissolution marks and minor damage, mostly indicative of chemical leaching. And the third group is highly fragmented shells. So these were qualitative groups based on how the forams were preserved. But we wanted to put numbers on this. So we wanted to count the relative percent of good, poor, and fragmented forams. So the metric had to be quick and unbiased. So we actually tried this on 300 forams, 500 forams, 700 forams, and 900 forams. And it turned out that 500 is a good number to count and calculate the relative abundance of these 3 categories. Another thing is that a proxy should be reproducible, which means if 2 people are working on the same sample and using the same proxy, they should arrive at the same relative percents. So there were 4 people, and we independently tried calculating based on our definition of these 3 categories, and we did arrive at the same percentages. So this is how we developed the proxy. So when we applied this, and this was mostly my work on Bidat, so I basically counted these 3 categories and calculated the relative percent abundance. And when we plotted it, it was really interesting to note that in the same interval of low magnetic susceptibility, there was a high abundance in terms of percentage of these dissolved shells, low percentage of well-preserved, and also an increase of fragmented shells. What was also interesting was in the same interval, he also had a preferential preservation of more robust shells of forams. So a logical double check was another section way away in Austria. This is called Gamsbach. And we pretty much found the same there of high fragmentation, low magnetic susceptibility, and concentration of more robust shells. So this was encouraging, but we have to play the devil's advocate before we make major interpretations. So the first is that if there was decreased calcification in planktic forams that flowed at the top of the ocean, and if the water dissolution was entirely restricted to the water column, then we should see enhanced test fragmentation, and the it's only the planktics that should show this corrosion and not the benthics that live at the bottom of the ocean. So the double check would be to see if benthics show a similar fragmentation index or not, and also to see if there are biological signals of this calcification crisis in planktics. The other point was the concentration of robust shells, and the easiest way to check this was to experimentally simulate a dissolution event and see if the same thing happens in the lab as we see in the sediment record. The next was what if these preservational conditions are purely sediment transport and reworking? In this case, I think both the planktics that live at the surface of the ocean and benthics that live at the bottom of the ocean would just be treated as sediment particles and should both show the same kind of wear and tear, which is not something that we saw in the samples. And another point is that local conditions affect a lot in terms of the preservation. And that's why we needed to check other distal K-Pg boundary sites to see if the same trend occurs everywhere, so that we can actually talk about a globally spread out crisis. So using this checklist, some of our next steps were to check for biological effects. This is again Bidarth, the low magnetic susceptibility interval with high fragmentation. This is the work of one of my master's students, Shraddha, and preliminary results from different species of different morphologies show that there is actually a decreasing trend in their shell sizes. As you can see, this is just pictorial representation for you. So there is a decreasing trend in their average sizes. And so this would basically be intraspecific dwarfing. Another thing that we see is exactly in the low benchmark high fragmentation index interval, we see an abundance of these disaster opportunist triserial guys. Just to give you a comparison, the larger forams require 300 times the calcium carbonate as required by these small guys to build their shells. So no wonder in such a crisis, you have more of these guys and very few of these. So this was evidence for interspecific, preliminary evidence for interspecific and intraspecific dwarfing. We have better results for this, which hopefully we will submit by this summer. The next was experimentally dissolving forams, and we basically got inspired by Nguyen et al. And they used sodium acetate and NaOH to basically have different pHs and track dissolution. This was a little difficult and had a lot of initial failures because we were not tracking the same shells dissolving through time. And there were problems with the buffer. Every 2 days, we had to change the buffer. In the process of changing the buffer, we were destroying the shells, which are very vulnerable to dissolution already. So this was not a realistic experiment. And in no way is pH 5 a realistic surface ocean pH estimate. So throughout the Phanerozoic, the pH has never gone below, let's say, 7.5. So we needed an experiment with realistic pHs and realistic exposure times, which is why we devised the next round of experiments. This had the good, the bad, and the ugly now into a more refined scale of preservation state. And we gave them scores or numbers. And what we changed was the experimental method, where we shifted to an organic buffer. This buffer holds good for months, and it operates over a pH range of 6.5 to 8, which is perfect for us. And we used realistic pH ranges, especially between 7 and 8. So the most important findings were, yes, low pHs resulted in poorer score or poorer preservation. But most importantly, it showed us that realistic estimates of surface ocean low pH conditions did actually result in poorer preservation. And it operated over short enough time scales, which would affect the planktic forams while they are floating in the surface ocean. Another important finding was that once the shells were exposed to low pH and then exposed to high energy conditions, it did result in higher fragmentation, which builds back into the hypothesis that I just explained. So the fragmentation index really works. Now, the third angle to our checklist of things to double check were benthic fragmentation. And as you can see, benthics don't really show much fragmentation here compared to the planktics. So that was also important to constrain the problem to the water column. By now, we also had another proxy for volcanism, which is Mercury, and I will talk about it in a bit. The next point was geographic spread of this event, the acidification event. And we tested a couple of sites from the Texas, the Southern Ocean, Atlantic, and the results are pretty encouraging, although I never really published this. We do see an increased dissolution in the last couple of centimeters below the K-Pg boundary. So taking stock of the situation, yes, there is no bottom water acidification event because the benthic fragmentation index does not show much. There is preferential preservation in the rock record, which can be simulated experimentally, and that kind of lends some substance to that idea. Fragmentation index actually works, experimentally speaking, and that's very encouraging. And of course, there is a geographic spread of this event just before the K-Pg boundary. Also, there is independent biological signal for a calcification crisis in the final 60 centimeters of the late Maastrichtian or the Cretaceous in Bidat. So something fishy was going on. And the next question is, when did it begin? How long did it last? And what are the implications for the K-Pg boundary mass extinction? This is a grand figure from the recent paper by Keller et al. 2020. Shows a lot of faunal changes and climate change associated with Deccan volcanism. It also shows Mercury peaks, which are a good proxy for volcanic events. Based on the peaks, eruptive events have been recognized, and this is all data from LS, Tunisia, which is the most expanded and well-preserved section for the Cretaceous-Palaeogene transition known so far. We also have cyclostratigraphy to calibrate the durations, and here are the dates for Deccan traps. So what I would like to orient you with is that is where the high fragmentation interval correlates to, and this is the final acidification event preceding the K-Pg boundary. Now, this equates to 2 precession cycles, which would be between 40 to 50,000 years. If you take 20,000 years here, it would be 40,000 years. If you take 23, then it would be 52,000 years. So this is 40 to 50,000 years just before the K-Pg boundary. So how do we explain this? If we go by the impact is the K-Pg and K-Pg is the impact scenario, then it post-dates this whole crisis event. So I'm not sure how you can explain the event being caused by the impact. If you go by the other scenario where the impact spherules were found at the base of zone CF1 in the Caribbean and Mexico, so this scenario would put the impact about more than 200,000 years before the mass extinction, and still can't explain how and what that has to do with this event just before the K-Pg boundary. Also, impact, the consequences of an impact would probably not last more than a couple of years to a couple of decades, maximum centuries. So that doesn't even show up in the resolution of this record shown in this figure. The other angle is these eruptions or the eruptive events identified. And what's most interesting is that in that interval with the calcification crisis, you have multiple hits of volcanism identified as these peaks of Mercury. And that seems to fit better with the duration, holding the oceans at low pH conditions for 40 to 50,000 years with multiple eruptive events. So I think I will wind up at this point and thank all my collaborators. Of course, my advisor, Professor Goethe Keller, Dr. Paula Matteo, Dr. Thierry Adat, Dr. Hassan, Martin, Jorge Spangenberg, Eric Font, Sigala Bramovic, and especially to my students who are currently working diligently to get more data to talk about the biological aspects of ocean acidification. Thank you.
Thank you, Jahnavi. That was excellent. So if people have questions, please either unmute and speak up or put them in the chat window so that I can read it out for Jahnavi. I think we already have our first question. The Deccan traps are basically opposite to the Yucatan on the globe. Could the bolide have triggered the Indian event?
There is a lot of discussion about this. Actually, it's not antipodal if you take the globe and check. It's at some angle, and it's not exactly diametrically opposite. And I think there are people who have run model simulations to see how shock waves from the impact can affect volcanism. In fact, more recently, there is a hypothesis that impact might have caused a state shift in the Deccan volcanism. I don't have any expert comments on this, but I'm not sure how that would work. I mean, just trying to process. I'm not too sure if they can be related, but I'm not an expert, so I wouldn't. Yeah, any others?
While we're waiting, I have a question. Why is Mercury thought to be a signature for volcanic events in sedimentary section?
Very interesting. Long story. Long story short, all the mercury that comes into the atmosphere is nascent mercury and is introduced by volcanic eruptions. It has a couple of years of residence time, and it's going to react with, let's say, sulfur oxides or chlorides and rain out and basically get sequestered with organic matter and get deposited. And of course, the oceans have a lot of sequestered mercury, but most of it that's coming in is with volcanic exhaust. So back to your question, I think this was when I was doing my PhD. There was a lot of excitement about the Permo-Triassic mass extinction having a mercury anomaly associated with it, and that was basically proposed as a marker for volcanism. And after that, I think pretty much every faunal turnover event that's been linked with a volcanic event has shown mercury peaks. And it's basically, again, of course, it has its shortcomings because you can have other processes to sequester mercury which may not be related to volcanism. However, you can always normalize it with the organic carbon or the clay content or pyrite or whatever is the process that might sequester it, and you still have these peaks associated with volcanic events. So also recent volcanic events like Tambora, and they also have association with mercury, increased mercury content in the atmosphere.
Okay.
So
I think we have a question from the audience. I see a raised hand, so please unmute and speak up.
Excuse me, ma'am. I'm audible?
Yeah. Yes.
Yeah, see, when you use fragility index as a proxy, what is the significance here? Like tropical sea surface temperature in the Cretaceous period, that will exceed, right? More than 35 or 32, right? So will it affect in the fragility index and or local perturbance, or I mean, just I want to know sea surface
I think
temperature
it's
in the region. Yeah.
Yeah, I'm not too sure if it has a direct link with sea surface temperature per se, but you're right in that fragmentation index has a lot of variables controlling it.
Yeah,
So
because the value you have
it's
given.
not just, yeah. So it's not,
Along the.
so clearly it's not, yeah, go ahead.
Sorry, ma'am. Carry on. Continue.
Yeah. So what I'm saying is that it's not, it cannot be a sole proxy for acidification, because a lot of things affect the preservation of forams. It could be local conditions, it could be the morphology of the foram, it could be a lot of things. However, that's why we are trying to couple it with other proxies and see if there was an acidification event. Back to your question about sea surface temperature, I can't think of a direct relationship with temperature, but yes, if global warming is associated with surface acidification, then it would affect the preservation.
Okay.
It's
And
complicated. It's not that straightforward. Yeah.
yeah. And this fragility index, whatever you're calculating, like even end of the Cretaceous, even many authors they have suggested asteroid impact and everything. So will it like so how you define how you connect with this asteroid impact with our results?
Very good question. I think I gave you two scenarios of the timings that people have proposed for the impact. And in both scenarios, it does not affect the acidification interval that I was talking about. However, there are publications that discuss the link between asteroid impact and an acidification event. Let me just, I did put a couple of slides for that. And if you look at this particular paper, I think you could go back and read this. This is Tyrrell et al. From PNAS 2015. Yeah. So they actually model different scenarios with which you can have acidification related to an impact. And if you see the x-axis of time after impact, in any scenario, it does not last beyond, let's say, tens of years. Okay, so if there is one impact and it has any sized impact, the effects of it on the oceanography would not last beyond, let's say, decades, maximum, let's say, a couple of centuries. Now, if you go back to this figure and the timeline that I showed you, even with the highest resolution, we are still talking about two samples apart by a couple of hundreds of years. So there is no way you can have effects of one impact show up on this record at all.
Okay.
So one of the propositions is that the KT boundary itself, the major extinction there itself is because of acidification due to impact. But then if you go back to these models, if the effects don't stick around for more than a couple of decades, then you have to kill all the organisms within those few decades. Now, there are other papers that show you that organisms have a tendency to adapt to these situations. So the initial response is not just extinction. First, they will try to adjust by, I don't know, dwarfing, trying to create microenvironments within their cells to still precipitate calcium carbonate. Yes, it is stress, but they will still try to adjust. Only in an extreme case scenario, if the pH drop is very drastic, yes, you can kill them. But there is not much discussing that. I'm not sure if that helped.
So we have a couple more questions from the audience. Here is one. When you look at forams at the KT boundary worldwide, are there any regional trends that you detected? And if so, what would you attribute those differences to?
Regional trends in terms of similarities or differences. So it's complicated because if you think of each depositional setting, it has its own variables. So trying to get a trend which goes across, let's say, the Paleo-Texas, so it's not a very easy thing because you'll have a lot of local influences, and the database basically consists of sections that were, I don't know, shelf, slope, different depositional environments, and each will have their own variables controlling preservation, if we're talking about preservation. So what has actually worked is the magnetic susceptibility, low magnetic susceptibility interval and Mercury. These have really worked very well. And surprisingly, if you're talking about Mercury, there is not much of a peak if you're closer to Deccan, but you do see good peaks when you go to distal sections in the Tethys, if I'm not wrong. Okay. Magnetic susceptibility also shows a distinct interval in slope settings, section from slope settings. So you don't want extremely shallow marine sections because they have a lot of noise because of high sedimentation rates, and these signals get diluted. You also don't want a very deep marine setting where you have hiatuses and condensed sediments. So you want something of a balance, and slope settings seem to be the thing. So Bidarth, for example, is approximately 1,000-meter paleo depth. Yeah. So that, and regional trends, well, what do you exactly mean by regional trends? In terms of fauna, in terms of geochemical proxies? Fauna, not really. Again, it depends on the local, each place has its own. Some places, the extinction looks very sudden because the sedimentation rate is low, and that kind of compresses the data set and makes things look more sudden. In more shallow settings where there is a higher sedimentation rate, there's a better resolution of these faunal events. So it's not very easy to compare between sections.
Can you?
There is another question. Yeah.
Can you determine the sedimentation rate independent of the foraminifera?
How do your findings for the K-Pg boundary suggesting species extinction being caused by volcanism correlate with other extinction events? Again, it's not straightforward because what's that? I don't know. Yeah. So it's not easy because the kind of organisms that went extinct in different extinction events are also very different. So how will you compare them? If you're talking about planktic forams, you can possibly compare with, no. So planktic forams evolve in the mid-Jurassic. So the only mass extinction they witnessed is the End-Cretaceous. Of course, there are other turnover events in the Cretaceous, and they do get affected. Not sure if that answers your question. But yes, you can have metrics defining how many species of what groups went extinct in what time window, and those numbers can help you compare between different mass extinctions. Yeah.
It looks like someone got the timing wrong. Are there more questions from the audience? All right, if not, let's thank our speaker. Thank you for such an excellent talk, Jahnavi.
Thank you for inviting me.